Today, I would like to discuss diversity in our immune system. Diversity is actually deeply embedded in our immune system because of evolution. Now, the important thing to understand about evolution is that evolution is always a competition. For example, you might have a competition between predators and prey. Prey might evolve to get faster, such as a gazelle, while predators will also evolve to get faster, such as a cheetah. And essentially, this race to get faster tells you who's going to live and who's going to die. Think, for example, of a population of gazelle. The slowest gazelle is the one that's going to be eaten by the cheetah. So only the fastest gazelle survive. Population becomes faster on average. And at the same time, if you look at the cheetahs, only the fastest cheetah is going to catch the gazelle. So the fastest cheetahs survive, the population gets faster on average. And as the population of gazelles and cheetahs both get faster, there's a race, and both of them become faster and faster over time. It's a form of specialization. Of course, in a predator-prey relationship, it's not just speed. Different pairs of prey and predators will be specializing on different aspects. It could be speed, it could be strength, it could be intelligence. Generally speaking, it is very positive for a predator and a prey to specialize on one particular aspect of the competition, rather than trying to do all aspects at the same time. Now, this becomes quite different when you think about the predator-prey relationship between pathogens and humans. And the reason why this is different is because we're not really talking about a relationship between the pathogen and the human. We're talking about a relationship between the pathogen and our immune system. The problem is that while every generation, both sides can get stronger, there's a massive asymmetry in the generation time. So for humans, a generation will be 20 years or so, while in bacteria, a generation can be 20 minutes. This means that the bacteria or other pathogens can really evolve much, much faster than humans. So if we just have a simple predator-prey relationship, those pathogens should be able to very quickly out-evolve us and our immune system. In fact, if we all responded the same, pathogens would win. So instead of looking at the gazelle and the cheetah, we can think of the bacteria and our immune system. And as the bacteria moves from person to person to person, they're going to become stronger and stronger and stronger in their ability to overcome our immune system. This means very quickly we're going to be able to have pathogens that can just wipe out humans. The best advantage we have for dealing with this situation is that we actually have multiple different immune systems. We don't just have one immune system. So you can think of the different types of immune systems that we have. They're all present in all of us, more or less. And different people will be prone to activate one type of the immune system or another type of the immune system. And we can consider this propensity to activate one type of the immune system or another as where we lie in immune space. So let's think of the three basic types of immune responses that we'll have. The first is type 1 inflammation. Immunologists are simple people. Second, type 2 inflammation. I think you're getting the trend here. And the third, type 17. Okay, immunologists don't always make complete sense. But let's just consider those three different types of inflammation. And this checkerboard will be a visual representation of where different individuals lie in terms of their propensity to respond with type 1 or type 2 or type 17 inflammation. So individual who's blue might be more prone to respond with type 1, individual who's red might be more prone to respond with type 2. Now, if we think about this race between the cheetah and the gazelle, but now with the pathogen and the human, we think that the pathogen can be specializing towards, say, type 17 inflammation. And this might be fine if it passes from an individual who's type 17 biased to an individual who's type 17 biased, and so forth. But if that pathogen then hits an individual who's type 1 biased and an individual who's type 2 biased, then that specialization that the pathogen has embarked upon now becomes detrimental, and the pathogen doesn't do very well. So by having a diverse population, we win. It's actually better for us if those people around us have a different immune system to us. We want everyone to be as different as possible in their immune system. Essentially, the more diverse we are as a species in our immune system, the less pathogens are able to specialize in attacking us, and the better we are in terms of everyone's health. Of course, immune diversity does cause problems as well. Those types of immune responses, type 1, type 2, type 17, they can all cause problems. And this is why some people are going to be prone to autoimmune diseases. Some people are going to be more prone to having long-term infections. Some people get allergies. Some people get chronic inflammations, because all of these different immune responses can cause attacks on our body. And you might be really good at fighting off respiratory infections, but at the same time, you might be prone to having allergy. It's always going to be a trade-off in the immune system. So we know that immune diversity is important, but what actually controls immune diversity? What controls whether or not one person is Th1 biased or type 1 inflammation biased, another person is type 2 inflammation biased? Well, to answer this, we embarked on a project where we were measuring the immune system in more than 2,000 individuals. What we did is we took blood samples from all of these individuals. We then profiled their blood using some sophisticated techniques, and we basically were able to say for every individual how prone they were to responding with different types of immune response. And then we can ask some simple questions from this. For example, we can say, how stable are people's immune systems over time? So here we can say that the blue individual is type 1 biased, the red individual is type 2 biased. But is that true just for a snapshot? If it's true today, is it going to be true next month, next year? And we can ask this question and then answer it by looking at these individuals over and over again over the course of months or years. And essentially, we had two basic hypotheses. One possibility is that immune state variation is unstable. So if you look on one day or you look 2 months later, that individual might now have a completely different immune profile. The second possibility is that the immune variation is more or less stable. So if someone is type 1 immune biased on day one, they're going to be type 1 immune biased a year from now. So we had these two basic hypotheses. So this is the hypothesis for basically an unstable variation. You can see the red and the blue individuals are moving around in immunological space. One snapshot, you might say one is type 1 and one is type 2. Another snapshot, you'll say they're both type 2 or both type 1. And it's just going to go on. Whereas in this stable state, you can see there's a little bit of wobbling, but essentially that individual that's type 1 is staying type 1, and the individual who's type 2 is staying type 2, because they're staying in that same region of immunological space. So what we did is we essentially tested 200 individuals at 6-month intervals over the course of a couple of years. And the long story short is that more or less those individuals were stable in their variation. The ones who were type 1 biased, they basically stayed type 1 biased over the course of years. And those who are type 2 biased basically stayed type 2 biased over the course of years. The next question we want to ask is what happens after an infection? Because being stable while healthy is not the same thing as being stable after an infection. You could imagine that individuals could be stable, their immune system changes on an infection, and then they default back to their original stable state. So maybe this individual is type 1 biased, they have a response where they really get driven to do a type 2 response, but then they come back to type 1 afterwards. Now, the alternative hypothesis is that individuals actually settle into a new state. So here you can imagine there might be type 1 and type 2 biased originally, but given an infection that needs a type 17 response, they both respond with that type of infection, and then they more or less settle down in this new stable state. So what we wanted to do is to test this hypothesis with a genuine challenge infection. And so our challenge infection is severe untreated gastroenteritis. We called this study the Fun Holiday Study. Essentially, we had 50 travelers that came into the clinic. They were coming in for their routine travel vaccinations and travel advice, and they wanted to go to countries where there's a higher risk of gastroenteritis. We took a blood sample before, and we said, have fun on your holiday, try the street food, see what happens. And they came back afterwards and told us how they went. Did they have gastroenteritis? Were they healthy the whole time? And we took a blood sample at that end. And so we can see here the dots on the left. These are the dots that are before they went on holiday. Then there's the grey bars. This is the period where they were on holiday. And then some of these individuals had gastroenteritis. So you can see the second individual had a short period of gastroenteritis in their holiday. Further down, you can see some poor individual had gastroenteritis for 10 days of their 2-week holiday, which is really quite horrible. And in each case, after they came back, we were able to take a new sample and look at where their immune system was in that case. And essentially what we found is that regardless of whether or not an individual was healthy the whole time of their holiday, or if they went and had this acute gastroenteritis, their immune distance from where they started to where they ended is basically the same. So they had a very stable immune response, even if they had this big challenging infection in between. And so this gives us the answer that actually individuals have stable immune systems that are stable even on a challenge. That actually brings us to another question as to why they have such different baselines. Why is one individual type 1 biased and another individual type 17 biased? Part of the answer is genetics. So what we did next is we took 500 individuals, we calculated 100 immune parameters from their blood, and then we also analysed them for 1 million genetic variants. And we're able to see which immune variants changed with different genetic variants. Essentially, we discovered that there are novel genetic variants that control parts of our immune system. This is on the right called a Manhattan plot. Essentially, it's meant to show different genes controlling different immune profiles, different parts of the immune response. And so you can see a gene on chromosome 2, CD45RA, is able to control one particular immune correlate, another will control another, and so forth. It's called a Manhattan plot because it's meant to look like the skyline of Manhattan with all of these skyscrapers where you see a genetic signal. And we were able to find multiple different genetic linkages that control multiple different immune traits. And essentially these new discoveries, they control about 5% of the difference between different individuals. And we're also able to calculate that unknown genetics control another 25%. So overall, about 30% of the genetic difference, about 30% of the immunological difference from individual to individual is controlled by genetics. We're also able to find that age controls about another 5% of the variation. So of course, as you're getting older, your immune system is changing, and so you can predict that for about 5% of the immune changes that we see. What about the other 65% of variation? Well, here we're able to find that the environment plays a role. What we were able to do to get to this answer is look at 138 couples that were living with a child at home. And we then measured the similarity between their immune system. And so what we can see is for every individual here, we can have a female and a male. They're a cohabiting couple with a child at home. And you can see that these two individuals, they're quite different from most other individuals in terms of their immune profile, but they're closer to each other than they are to everyone else. This couple as well, they're a bit of outliers, but they're relatively similar to each other. And you'll see this repeating over and over again. These couples seem to be aligned fairly closely to each other. And we can measure this distance between the different couples, or we can take all the couples and then we can reassign them at random and see how different their immune system would be if they were just random pairs. And what we see is that the random couples actually have about twice as much immune variation as the real couples that we analysed. Now, ideally, we would be able to get all of these couples to get divorced, get remarried, and see if their immune system changes. But since that's not really possible, what we can say at this stage is that people who are living together and have a child together, they tend to have a much more similar immune system than we would expect by chance. How could this happen? Well, so what we would have is an individual who's type 1 and an individual who's type 2 biased. At some point, well, they meet, they fall in love, and their immune systems are becoming more and more similar to each other. So maybe they're now sort of in between where they were starting off. What could drive this effect though? Well, actually many different things are shared by couples that are environmental. Couples tend to eat the same food. They tend to have a more similar exercise regime. They tend to share similar stresses. They're sharing similar environmental exposures at home. And if we think about the bacteria that live in our body, there are literally trillions of bacteria that are living in our guts, living in our skin, and we're exchanging thousands of bacteria in a handshake and millions in a kiss. And these bacteria are also then able to shape our immune system. So the fact that people are living together and cohabiting together means that they're going to be sharing all of these environmental exposures, they're going to be sharing these bacteria, and that could account for why their immune systems are becoming more similar. Overall, though, there is still a lot that is unknown in terms of what controls the diversity of our immune system. As I mentioned, we know about 30% is genetic, and we actually know the genes of a fraction of this, maybe 5%. We know that there are a number of different causes that are environmental, age, sex, smoking, some infections. And then we know that there's a big extra environmental component because we can see it in cohabitation, but we don't really know which parts of the environment are driving which immune profiles. And this is really quite important because understanding how the environment is changing our immune system really could be the future of immune therapy. You could imagine a not too distant future where we can diagnose an immune problem before there's any clinical symptoms. We could say that this individual looks like they're on their way to developing a disease that is caused by type 2 inflammation. We could then use a targeted environmental change to correct the immune system, push them more from type 2 inflammation into type 1 inflammation. And that would prevent the disease from even starting. Now, of course, this really requires us to know how their immune system is biased and which different environmental exposures are required for that person to push their immune system back into balance. And critically, this needs to be based on science, not on pseudoscience. There are so many claims out there that this fruit juice or that herbal supplement are going to fix your immune system. The thing is, none of this is actually based on good, solid research of how those different ingredients are going to work on individuals and what effect that's going to have on their clinical outcome. So this is the future, but we're not there yet because the research just hasn't been done. With that, I'd like to finish up. I really want to thank a couple of key people in the lab. James Dooley, who set up this platform for measuring the immune system of different people. Vasiliki Lagu, who led on the work involving genetics. Josie Garcia Perez, who was really quite instrumental in this work. Anne Goris, our key clinical collaborator. Michelle Linterman and Ed Carr, who were involved in the study looking at the challenge of healthy individuals. And of course, science isn't free, so our funders, BBSRC, the ERC, and the VIB, and all of the wonderful people that I've had the opportunity to work with while trying to study human immune variation. Thank you.